Well, I'd like to thank Professor Orley and the organizing committee for the opportunity to come and speak at this wonderful meeting. It's really quite nice and excellent science. I get to see some of my friends and enjoy this lovely city, and the opportunity has been quite nice. I'm going to talk to you about an observation that was made actually about 60 years ago by Hans Selye, the famous Nobel-winning Hungarian physiologist, who observed that there were rapid effects of glucocorticoids that would occur within minutes in the vasculature. This obviously could not be explained by some process that involved transcription and then translation and then precise localization of the protein and function. And this really started the concept of rapid actions of steroid hormones. And I'm going to tell you about the nature of these steroid receptors and some biochemistry, but every time I do that, I will then move into a clinical application to help us understand where these extranuclear receptors play important roles in physiology and in either the production of or the opposition to pathological states. So let me start out by giving you a little bit of a biochemical overview. First of all, the membrane-associated steroid receptor pool is the classical nuclear receptor that traffics to the plasma membrane as a monomer. It rapidly engages ligand in terms of estradiol and dimerizes. Just like the nuclear receptor, dimerization is necessary for the activation of this receptor pool. Upon dimerization, it can contact G proteins like G-alpha-Q and G-alpha-S. And in a specific context, both cellular and in a physiological sense, different G protein subunits are activated. So this is a G protein coupled receptor, and it spans the membrane. Not like the adrenergic or dopaminergic or angiotensin receptor, but nevertheless, it does activate G proteins and signals in that fashion. So within seconds, let's see if I get my arrow here. Here we go. So within seconds, calcium is activated. Proximal kinases like PKC are activated. Cyclic nucleotides like cyclic AMP and GMP are generated. And then within minutes, there are signal cascades. And these kinase cascades ultimately affect existing proteins and modulate responses like proliferation, survival, and many other developmental aspects. Now, in hormone-responsive cancers, the membrane estrogen receptor actually crosstalks and activates growth factor receptors at the tyrosine kinases at the membrane, such as the EGF receptor and the IGF receptor. And in fact, if you either genetically delete or block the tyrosine kinase functions of these receptors, you lose the ability of estrogen to signal through the important downstream pathways that ultimately lead to cell proliferation and cell survival through nuclear effects. So this crosstalk is essential and creates opportunities both to understand and interrupt the effects of the membrane receptor pool in hormone-responsive cancers. So why do we study this? Is this just an interesting phenomenon, or is there a real application? So this is a panel of breast cancers from women with ER-positive cancers. So here's a very, very typical estrogen receptor positive cancer. And to remind you, the pathologist will designate that a cancer is ER positive or negative on the following basis. 10% or more of the cancer cells must show nuclear ER alpha. That's been the traditional concept for 40 years. And it is incredibly important because the therapy that a woman would get is predicated upon whether ER is present or not. So this woman who has a classical nuclear receptor abundant ER positive in the nuclear tumor would get localized therapy, that is a combination of limited surgery and radiation, and then would get an endocrine therapy, most often an aromatase inhibitor at this point in time. Now contrast this tumor to the following one in panel B, and you can see here, get this, okay. You can see here, the red arrow is pointed to a tumor cell that has a lot of abundant nuclear ER alpha, but look at the adjacent cell. This is a halo effect because this tumor cell has membrane ER alpha and no nuclear ER alpha. So there is heterogeneity of the tumor for where the ER is localized. And then we have tumors where the predominant amount of the tumor, as shown here in C and in F, is what I call a honeycomb pattern. The majority of the tumor cells have only membrane ER alpha. Now it's an ER positive tumor because about 10%, which is all you need in the nucleus, do have the nuclear form. But the majority of the tumor is actually at the plasma membrane. And to take that one step further, we've been looking at ER negative breast cancers in women who receive chemotherapy and not endocrine therapy. And we found about 20% of those have abundant membrane ER alpha, but no nuclear ER alpha. Would those women have benefited from endocrine therapy? So we are doing a large study in collaboration with Hoda and Tom Culver to correlate in 300 women's specimens the pattern of ER expression and a 15-year history of response to therapy and the natural history of the disease, so we can begin to answer these questions retrospectively and then prospectively test out whether some women should be getting endocrine therapy who are not currently. Now, how exactly does this receptor get to the plasma membrane? So we and other labs have spent a lot of time on this. I'm just going to give you some summary statements about this. So the receptor is made of, of course, the endoplasmic reticulum. It traffics to the Golgi, and there it either moves under chaperoning by heat shock protein 90 to the nucleus. About 85% of the receptors are in the nucleus. And then there's another population that is altered by palmitylation, and that's shown here. And palmitylation is putting a palmitic acid group on a cysteine, usually internally, and that causes the physical association of ER with the caveolin-1 protein. And this complex then moves to the plasma membrane, and here the caveolin-1 coat protein of the caveoli raft assembles a signaling complex. And it's different in different cell types and in different contexts, and that's where the specificity of signaling occurs. Now, once this is assembled, there's activation in this closed temporal space, and there's cross-activation of the receptors that I told you about in hormone-responsive cancers, downstream signaling to all the effects that we know promote carcinogenesis in uterine and breast cancer as but some examples. Now, we showed about a year ago that heat shock protein 27 is really important for the palmitylation process to drive this association and caveolin-1 transporting ER alpha to the plasma membrane. But the big hole in the story is, what is the nature of the enzymes that actually cause the palmitylation? So we've been investigating that, and we first defined what's called a palmitylation motif of 9 amino acids. This is where the palmitic acid is actually attached to this cysteine. But all these elements in red are important. And just to give you an example, here's a wild-type estrogen receptor. If you express this in a cell, you get dense nuclear ER alpha, you get plasma membrane localization shown in white, and you get this cytoplasmic pool stippled throughout the cytoplasm. But if you then mutate, for instance, the cysteine, the point mutant, here you lose only membrane estrogen receptor localization and function. So it doesn't rapidly activate signal transduction, but it does normally act in the nucleus to activate ERE responsive genes. So this gives us A, an example of what the receptor is doing, B, how it gets there, and C, an opportunity to selectively disrupt the membrane pool and ask what is its contribution either with or apart from the nuclear pool. And I'm going to come back to that in a mouse that we made and some of the important insights we've gained from that shortly. Now, this is not only applicable to the estrogen receptor, but the same palmitylation motifs shown here, we showed by mutational analysis, are critical to drive the androgen receptor to the plasma membrane, very important in prostate cancer and very under-investigated. In other words, what is the role of the membrane androgen receptor in promoting prostate cancer? Completely unanswered, but likely to be very important. And additionally, there is a pool of membrane PR at the plasma membrane of breast cancer, and it has an important role, and I'll show you that shortly. So what is the nature then of the palmitoyl transferases? And we've recently published work where we've isolated the PATs that actually put the palmitic acid on the steroid receptors. And they turn out to be two. One is DHHC7 and DHHC21. So there are 23 members of the family in mammals of these palmitoyl transferases. But there are hundreds of proteins that must get palmitoylated to drive those proteins to the membrane. And you're very familiar with many of these. SARC, RAF, many of your proto-oncogenes, tyrosine kinases, integral membrane proteins, they all must be modified by palmitylation to traffic to the membrane. And so for the estrogen receptor, and it turns out also for the androgen and progesterone receptor, these are the two, DHHC7 and DHHC21. Now this has allowed us to define an important new function of the membrane receptor, and that is the promotion of epigenetic signaling. From work in our lab and other labs, I'll make a conclusive statement. It is the membrane receptor pool that is absolutely essential to drive epigenetic modulation in response to sex steroids. Now, a really nice example of this was published by Cheryl Walker's group about a year ago in Molendo. And Cheryl's very interested in diethylstilbestrol. To remind you, diethylstilbestrol was given to pregnant women in the 1960s and 1970s to stabilize their pregnancy. And there were no effects on the pregnant mother-to-be, but in the female offspring, there have been lots and lots of problems discovered. Most importantly, a rare form of vaginal cancer occurs in 30% of the female offspring of women who took diethylstilbestrol for stabilization of their pregnancy. And no mutations have been found to explain this new cancer that arises from DES usage. So that, in a legacy effect, really suggests that there is an epigenetic program to all this. And what Cheryl showed was that DES can signal through the PI3 kinase AKT field to cause a phosphorylation of EZH2, which is a histone methyltransferase. This is an inhibitory phosphorylation that lifts the repressive methyl mark at histone 3 and lysine 27. As a result, the chromatin remodels in a productive way and genes are transcribed. So we wanted to know whether this was applicable in breast cancer. And our target was the progesterone receptor gene. So to remind you, one of the major functions of the estrogen receptor in promoting breast cancer is to upregulate the progesterone receptor gene so that now progestins can act. And the Women's Health Initiative told us that estrogen alone is not sufficient to promote breast cancer. It's the combination of estrogen and progesterone. So the ability of ER to upregulate PR is essential to the carcinogenic process. So we looked in our MCF7 cell line model, and we added estrogen, and you see an activation of the AKT kinase. This causes a phosphorylation of EZH2 at the inhibitory site, and as a result, the histone methyl mark at lysine 27 of histone 3, which is a repressive mark, is lifted, and as a result, the progesterone receptor gene comes up quite strongly. Now, what if we block the membrane localization by knocking down those two palmitoyl transferases using siRNA? And that's shown in this slide over here. And we take it a little further by using a PI3 kinase inhibitor for the pathway, and that's shown over here. So now we don't get nearly the same degree of PI3 kinase AKT activation. The repressive phosphorylation of EZH2 is diminished. The loss of that repressive methyl mark starts returning, and as a result, the progesterone receptor gene is not nearly as well induced. And here, if you block PI3 kinase signaling, you completely block the activation of the progesterone receptor gene. So in other words, you need the membrane receptor to condition the histone and chromatin environment to allow the nuclear receptor to upregulate, in this case, the progesterone receptor gene. Now, is another example to convince you that this is terribly important. And this is work from Miguel Biedo in Barcelona, who's worked on membrane PR signaling. And in a series of papers, this is but one that you see down here in the lower right corner, he showed that ER and PR form a complex at the membrane. They activate SARC, the tyrosine kinase that you know is a proto-oncogene. That causes ERK activation, and then a portion of ERK moves to the nucleus. It phosphorylates nuclear PR, which recruits a kinase called MSK1, and this complex then phosphorylates repressive chromatin complexing, removing it from the level of the gene promoter, and as a result, many genes can then productively be upregulated. And in a 2008 paper in Molecular Endocrinology, Miguel asked a simple question. In a gene array where PR in the nucleus is activating hundreds and hundreds of genes, how many require membrane PR signaling through ERK to upregulate those genes? And his conclusion, over 90%. So this is not a trivial interaction. This is really fundamental to the way that sex steroids regulate genes. Now I want to talk to you about another really interesting area. We're doing a lot of work in this. Sometimes I feel like I'm the voice in the wind, but others I think will join in as this work becomes more mature. And that is steroid receptors in the mitochondria. And there's obviously implications for a number of different things. And I'm going to talk about their role in breast cancer. So we started out this work in 2006, and we showed that in your usual breast cancer cell lines, there's dense nuclear ER alpha, there's this membrane localized pool that you can see here. It's discontinuous because it's actively cycling in and out of the plasma membrane. And if you come back 20 minutes later and take a picture of the same cell, you'll see a different pattern at the membrane as it moves in and out into endosomes and then recycles back out into the plasma membrane. Now, there is this pool that I'll call your attention to just outside the nucleus, perinuclear, here and here. And this lines up very nicely with a dye that stains the mitochondria called mitotracker. And in yellow, you can see that there is a pool then of ER alpha in the mitochondria. Even more amazing is ER beta. And in many cell lines that we've looked at and others have looked at, ER beta is predominantly extranuclear, and it is in the mitochondria. And here you can see ER beta staining is all perinuclear. You don't see it at the membrane, you don't see it in the nucleus. It lines up beautifully with the mitotracker dye, and the majority of it is here. What is it doing there? Well, first of all, what is the origin of that? Is this the mitochondrial receptor, the bona fide classical ER alpha and ER beta? So to do that experiment, we made endothelial cells from wild-type mice and double knockout ER alpha, ER beta mice to interrogate that question. So here's ER alpha distribution these are normal aortic endothelial cells. It's mainly densely in the nucleus. You can see that. Here's my arrow again. There's a small population in the mitochondria of ER alpha. ER beta has a very different distribution. Again, it's all perinuclear. See how different that is than ER alpha distribution? And it lines up beautifully with the mitotracker dye. Again, unmanipulated cells. These are right out of the aorta. The ER beta is mainly in the mitochondria. But if you look at the cells from the double knockout mice, you see no ER alpha anywhere or ER beta anywhere. So these are products of the same gene. It is the same protein, but it traffics to the mitochondria. So what is it doing there? Let's go back to the breast cancer model. Estrogen is a potent survival factor, unfortunately, in breast cancer. And so if you treat breast cancer cells with radiation or chemotherapy, our traditional modalities to treat this malignancy, they cause an apoptotic cell death. They are cytotoxic. And they do this by inducing reactive oxygen species, which activate death kinases, phosphorylate BACs, which moves to the mitochondria, opens up the pore, out come the apoptosome contents, which activate effector caspases and kill the breast cancer cells. This is the intrinsic pathway of apoptosis. Estrogen, unfortunately, binds ER beta in the mitochondria and blocks the reactive oxygen species formation. It does this by upregulating a very important enzyme in the mitochondria called manganese superoxide dismutase, the major defense that you have in all your cells against oxidant stress. Now, in a woman who's getting adjuvant therapy and is having terrible hot flashes, we would love to give her estrogen to take care of that problem. But you can see that since the very modalities you're using to treat the cancer depend on upregulating ROS and causing apoptosis, you can't block that by giving estrogen. So this is one reason that women unfortunately cannot be given a sex steroid replacement, although they may be suffering from the effects of estrogen deprivation. Now, we were very interested more recently in unpublished work that we've just submitted to look at whether tamoxifen sensitivity or resistance may be determined by this feature. And in fact, we find that where tamoxifen works effectively, it is cytotoxic, and it works just like radiation or chemotherapy to upregulate reactive oxygen species, engage the intrinsic apoptotic pathway, and kill the cancer cells. And it does this by down-regulating manganese superoxide dismutase at both the gene, the protein, and most importantly, at the enzymatic activity level. In tamoxifen-resistant tumors, it's the opposite. Tamoxifen engages ER-beta as an agonist, down-regulates ROS by upregulating MNSOD, the apoptosis never occurs, the cells thrive, and we've been growing these tumors in nude mice with tamoxifen pellets in place to develop a model to see if we could reverse the tamoxifen resistance and restore tamoxifen sensitivity. And this has not been done in any in vivo model. And I'll show you our latest data on how we were able to accomplish this. So first, just to remind you, the reactive oxygen species are progressed. This is very DNA damaging. This is superoxide. It can be converted to peroxynitrite in collaboration with nitric oxide. This is also very DNA damaging. But here, manganese superoxide dismutase takes the superoxide to hydrogen peroxide. This is a much less damaging ROS. And then it's further broken down to carbon dioxide and water by catalase. These are your major defenders in your mitochondria against oxygen stress. So what we did was we grew up tamoxifen-resistant tumors in nude mice. So this is at 5 weeks. So this is one flank where we injected. Remember, this mouse has a tamoxifen pellet, which gives you very high levels, and yet the tumors grow quite nicely, as you can see. So we put it into both flanks, and then we injected at 5 weeks. Here's growth to 5 weeks. At 5 weeks, we injected either a control lentivirus shRNA or a lentivirus shRNA to knock down MNSOD, because that's the linchpin. We want to get the levels low so the ROS go up, induce apoptosis, and kill the tumor. At least that's the concept. And here's the proof. At 8 weeks, you can barely see any tumor over here in the lentivirus MNSOD injected tumors, but yet this tumor continues to grow. So this is the control tumor, continues to grow. This one undergoes strong apoptosis. If you go out to 10 weeks, there's basically no tumor left. So we can restore a tamoxifen-resistant tumor to tamoxifen sensitivity by targeting MNSOD in the tumor itself. And here's a little more data on that. These are comparisons at 8 weeks. If you go out to 10 weeks, you hardly see any tumor whatsoever. This is annexin staining. You can see that this is a tumor that even at 8 weeks is still undergoing apoptosis, as reflected by the annexin staining. And then also we got the MNSOD knockdown by this strategy and caspase-7 cleavage, which is indicative of ongoing cell death. All right, well, now I want to talk to you about another line of work, again, a paper that we've just submitted for publication. And this is a surprising effect that we found on the ability of estrogen from the membrane receptor to regulate lipid metabolism, and the lessons we've learned in normal mice and in hormone-responsive cancers. So to do this, we made a mouse, a transgenic mouse. I'm not going to go through this, but let me just tell you what it is. It is the Pierre Chambon ER-alpha total knockout mouse with the ligand binding domain of ER-alpha expressed in the mouse cells, all the cells, and targeted exclusively to the plasma membrane. So let me show you what that looks like. And we took liver, I can't go into this, but that liver is actually a very important target for estrogen action. All right, so this is, these are liver cells isolated from total knockout mice. This is just some background staining, non-specific staining. There is no effect of estrogen in this mouse, and you would expect that. Now here is the wild type mouse. There's dense nuclear ER alpha. There is membrane localized ER alpha. And then here is our membrane-only estrogen receptor mouse. And as you can see, other than the non-specific staining, all you see here is membrane-localized E domain of ER alpha. And signal transduction shown here, this is ERK-MAP kinase, is exactly the same in wild-type and membrane-only mice and absent in the total knockout mouse. And that's true for PI3 kinase, AKT activation. It's the same in both the models, but absent in the ERCO. Now, knowing that, we can now do an in vivo study to try and ask the question, what is the importance? And what we're really interested in is comparing wild type and membrane-only mouse, because if they're similar in some aspect, you would have to argue it's got to be coming from the membrane because there's no nuclear receptor in our membrane-only mouse. Yet the effects are going to be similar, and then of course absent in the total knockout. That's the plan. So we ovarectomized the mice. We introduced injections of a ER alpha agonist at a biologically relevant concentration for 3 days. We took out the liver, we did DNA microarrays, and we wanted to see what genes were regulated comparably in wild type and mower, but absent in ERCO, because that would argue it had to be from signaling from the membrane. What did we find? A lot of lessons. The first lesson, just looking at repressed genes, 650 genes are repressed in wild type, only 53 in membrane only. So you need the nuclear ER alpha for the ability of estrogen to repress genes in hepatocytes. Lesson number one. Lesson number two, there are 32 genes that are comparably regulated in the membrane only and wild type and completely absent from regulation standpoint in the ERCO. What are those genes? And they fall out into the lipid synthesis pathways. 7 of them are in the cholesterol synthesis pathway, and another 10 are in fatty acid and triglyceride synthesis. So we wanted to investigate this further, obviously, because all the key genes are here. The transcription factor, SREBP1, which is the transcription factor for all the genes in the lipid synthetic pathways. And not only in liver, but in fat and other areas where cholesterol and fatty acids are made, that gene is regulated by estrogen signaling from the membrane. Also, the rate-limiting enzyme HMG-CoA reductase for cholesterol synthesis is regulated. We validated this by the qPCR. Here's an example in the liver material from the wild-type mouse, suppression by PPT, and then the membrane-only mouse. And importantly, the transcription factor is comparably downregulated in those two mice and not touched at all in the ERCO mouse. We looked at the cholesterol content in the liver. We found very strong suppression of cholesterol content. Again, comparably in these two mouse, but absent in this mouse, the ERCO. And we looked at insulin stimulation in hepatocytes of cholesterol synthesis. Insulin is a very strong stimulus, very well known and well clarified stimulus. And in the wild type mouse and in the membrane only mouse, there's strong stimulation, and again, almost complete suppression and comparable suppression of cholesterol synthesis in the hepatocytes from wild type and mower mice. Well, I don't have time to go through all the details, so I'm going to cut to a summary slide and tell you what we found. It's very interesting. So normal cholesterol synthesis occurs when the SREBP transcription factor is chaperoned from the endoplasmic reticulum to the Golgi apparatus. And there it meets two proteases, site 1 and site 2 protease. It's cleaved, it becomes an N-truncated form, and this is the form that moves to the nucleus, binds to the promoters of all the lipid synthesis genes, and activates them. And what we found is that estrogen signaling through AMP kinase and ERK phosphorylate SREBP and prevent the physical association of these proteases with SREBP. Thus, it cannot be cleaved, and thus it is retained in cytoplasm, never goes to the nucleus, and the genes never get transcribed, and cholesterol is never synthesized. And that's the mechanism involved. And it emanates entirely from the membrane receptor signaling. So what are the conclusions? First, something that may be a bit dogma shaking for the nuclear receptor field, you can get gene regulation and it doesn't require the nuclear receptor for a subset of genes. 2, the membrane ER can suppress cholesterol synthesis, so there's a metabolic phenotype, and this occurs in the absence of the nuclear estrogen receptor, and it impacts bile acid synthesis, overall cholesterol homeostasis, and fat in the liver. Now, this also occurs in hepatocellular carcinoma, which estrogen suppresses quite nicely, shown by the work of Michael Caron. And in humans, males have 6-fold the amount of hepatocellular carcinoma that women have, and there's good data that estrogen prevents the inflammatory progression of chronic liver disease to fibrosis, fibronodular cirrhosis, post-necrotic cirrhosis, and then hepatocellular carcinoma. Data is very good, both in humans and in animal models. And the ability of estrogen to suppress cholesterol, fatty acid, and triglyceride synthesis, which any tumor needs to make cell membranes and proliferate, is probably an important mechanism whereby estrogen contributes to the suppression of hepatocellular carcinoma. Now, on the other hand, we have another paper we just submitted that in breast cancer, estrogen also activates AMP kinase, but this is a compensatory function which activates the mitochondrial pathways and promotes fatty acid synthesis, because there, estrogen is promoting cancer, and this impacts cell proliferation and the survival of breast cancer. So here, the same pathway, but utilized for slightly different purposes, but with big impacts on hormone-responsive cancers. Now, for the last part of my talk, I wanted to focus on the cardiovascular system, because we're very excited about this work. For our own personal work that I'll show you, this is the work that is most ready to go into translational trials, and we're working with a company to put it together a translational trial in women, and I'll tell you about that shortly. So the first thing we did a few years ago was look at estrogen-induced vasodilation, and this was a collaboration with Ghassan Kassab, a microcirculation scientist at UC Irvine. And Ghassan, his lab puts in these microcatheters into the carotid or femoral artery of mice. And then he can do pressure measurements and diameter measurements of the artery. And so if substance is a vasodilator, the diameter enlarges. If it's a vasoconstrictor, it shrinks. And he can also measure stress strain calculations against the arterial wall that damage the artery and lead to accelerated atherosclerosis. And estrogen strongly blocks those stress strain forces against the arterial wall, and within a minute of infusing estrogen into the catheter, we see a vasodilation. How does this work? This is entirely from in vivo work. The ER alpha, ER beta heterodimer responds to estrogen by upregulating signal transduction to ERK and PI3 kinase. This phosphorylates and activates the eNOS enzyme, which produces nitric oxide. And if you do this experiment in the eNOS knockout mouse, there's no vasodilatory effect of estrogen. Nitric oxide is the single most important molecule in your arteries for vascular health. And the loss of nitric oxide production portends a rapid acceleration in arteriosclerosis and other forms of arterial damage. Nitric oxide then moves from the endothelial cell to the adjacent vascular smooth muscle layer, causes a relaxation of the smooth muscle layer, and arterial dilation occurs. Now, after we published this, about a year later, a group did a study in young women. They put an estrogen patch on the forearm, and they measured arterial resistance non-invasively within 10 minutes of putting on the patch, and they showed relaxation and dilation. So this is applicable to humans as well as mouse models, and this is the mechanism involved. Now, recent work from Phil Scholl's group has shown another arterial injury that estrogen nicely prevents, and it comes from the membrane receptor signaling. So Phil has published a number of papers, as have others in the cardiovascular area, where they do angioplasty. So to remind you, when someone has a blocked coronary artery, they get an angioplasty, a stent is put in to keep the artery open, and the person then is relieved from their symptoms of chest pain and angina. So if you do this in a mouse, you get tremendous vascular smooth muscle hyperplasia and migration to the intimal lining of the vessel. But if you do the angioplasty in the presence of estrogen, you block it substantially. And here's the intimal area without estrogen, and then if you add estrogen, you dramatically block that. Now, John Katz-Neuenbogen has made an estrogen compound which is called the EDC, or dendrimeric conjugate. And this takes estrogen and links it to long-chain fatty acids that keep it from entering into the cell. It never gets to the nuclear receptor. It only binds to the membrane receptor. So Phil used this compound, and he showed that the estrogen effect of the EDC compound is quite potent, just about as good as estrogen in preventing the acute arterial injury. And he did this in a second model, and this is the paper published in 2010. And he did it in the second model of acute electrical injury, which damages the endothelial lining. And again, estrogen was very, very powerful in preventing the injury, and it did it through signaling from the membrane estrogen receptor. Well, we've been very interested most recently in prevention of cardiac hypertrophy. So let me show you what this looks like. This is a normal female mouse heart, shown here, unstressed. And this is a female mouse heart from a mouse that's been ovariectomized, but not stressed in any way. And you can see the hearts look fairly similar at a gross level. Now, if you infuse angiotensin over 21 days to promote cardiac hypertrophy, and remember, angiotensin is the single most common cause of human cardiac hypertrophy and then progression to heart failure in humans. So this is a very relevant model that the cardiovascular scientists use. And again, to remind you that heart failure is the #1 cause of death. I'm sure it's the way it is in Israel, it is in the United States certainly as well. Now, if you do the infusion, you get cardiac hypertrophy in the intact mouse. But if you do the infusion in the presence of ovariectomy, look at the size of this ventricle. It's huge, tremendous cardiac hypertrophy. And the only difference between this mouse and this mouse is endogenous steroids from the ovary. And to show you that the protection is by estrogen, if you take this mouse and you add back physiological amounts of estrogen, you markedly downregulate the hypertrophy by about 65%. Let me show you what that looks like in transverse section. Here's the left ventricle and right ventricle of the heart, the interventricular septum. If you infuse angiotensin, you close over the left ventricle, but if you give estrogen, you block that hypertrophy. The blockage occurs in wild-type mice. It occurs in the ER-alpha knockout mouse, but the protection does not occur in ER-beta knockout mice. And 4 groups have now shown that it's ER-beta that's the player in the prevention of the hypertrophy. And how does that work at a cellular molecular level? And so from a series of papers, we showed that estrogen action can block angiotensin signaling to hypertrophy. So angiotensin activates a calcium-dependent activation of a phosphatase called calcineurin. This dephosphorylates a family of transcription factors, which upon dephosphorylation, they move to the nucleus, they collaborate with other transcription factors, and all the genes that produce cardiac hypertrophy and rearrangement of the basic structure of the heart cells and fibrosis of the heart, which impedes its function, all of this is attributed to this pathway. And Eric Olson, who's in the National Academy of Sciences for this work at UT Southwestern, has done the elegant work to define this important pathway. We showed that estrogen-engaging ER-beta activates through PI3 kinase signaling a gene called MCYP1. The protein product binds and blocks the phosphatase activity. As a result, there is no dephosphorylation, and as a result, the hypertrophy is dramatically lessened because this transcription factor never moves to the nucleus. We also looked at cardiac fibrosis. When you transition from hypertrophy to heart failure, fibrosis is a very important part of the process. So you want to prevent this, very important. So here is angiotensin-induced fibrosis. You can see it, the blue staining is Masson stain. It stains for collagen deposition, which is fibrosis. And you can see angiotensin causes tremendous upregulation of fibrosis of the heart. But estrogen is a very potent antifibrotic. 90% of the fibrosis is blocked in vivo. I know of nothing that is this antifibrotic, and believe me, many people have looked. So it happens in wild-type mice and in ER-alpha knockout mice, but it fails to happen in ER-beta knockout mice. Again, ER-beta is the player. And how does that work? The key fibrotic factors, endothelin and angiotensin 2, induce a TGF-beta dependent activation of junk, phosphorylation of the transcription factors that move into the nucleus of the myofibroblast. So TGF-beta changes the fibroblast into a myofibroblast, and this is the form that makes the collagen that gives you fibrosis, along with the other very important genes like alpha-synuclein, fibronectin, and vimentin. So how does estrogen working through ER-beta block that? It blocks TGF-beta formation. It signals through cyclic AMP and pKa to block junk activation. The SMAD transcription factors are not phosphorylated and they're retained in cytoplasm. And this summarizes those two pathways. So I've now given you 3 in vivo models where estrogen prevents pathology, 1 in the liver and 2 in the heart. And in each case, signaling from the membrane receptor causes a post-translational modification of the transcription factor, retaining it in cytoplasm. It never moves to the nucleus and never transcribes the pathological genes. And that's how estrogen prevents the pathology. So it's a novel concept of gene repression and prevention of pathology involving membrane receptor signaling. Is this only in the heart? Jan-Åke Gustafsson, who discovered ER-beta in his ER-beta knockout mouse, showed that unprovoked, that mouse has pulmonary fibrosis. So if you look at the end of the lung, this is the terminal alveolus, which is boxed over here, blown up over here. This kind of onion skin pattern shown by the yellow arrows is fibrosis. And this causes hypoxia because you can't exchange oxygen in this mouse once you have fibrosis. So ER-beta is defending against pulmonary fibrosis. And for the clinicians in the audience, you know that pulmonary fibrosis is a terribly difficult disease to treat. Perhaps an ER-beta agonist would be useful, a hypothesis to test. And again, as I mentioned, in the liver, the progression of inflammatory liver disease to fibrosis in women who have chronic hepatitis C infection is long-term benefited by estrogen exposure. And then, as I mentioned also, the progression to hepatocellular carcinoma. So this is applicable, the anti-fibrotic properties from membrane receptor signaling in many of our organs and prevention of pathology. Well, I just want to tell you that this field has advanced because there's been the seminal work of a group of scientists working in the estrogen receptor field, in the progesterone receptor field, working on the androgen receptor at the plasma membrane, the thyroid receptor at the membrane, both the mineralocorticoid receptor and the vitamin D receptor signal from the membrane. And understanding this in all these different areas has allowed us to find commonalities of function and ultimate the downstream effects that affect normal physiology and pathophysiology. In my lab, Manaz Razandi and Ali Pedram have been my collaborators for 20 years, and they do most of the work that I've shown you here today and deserve the credit. And this is Claire Zago, who started this all in the 1960s and 1970s with seminal papers showing rapid actions of estrogen that occurred in seconds and occurred from binding some undefined binding protein outside the nucleus. And this was published in Nature and received a lot of play and a lot of resistance, and any new concept will receive resistance. Claire sends me emails every now and then. She's in her mid-80s and still vibrant and engaged, and is a delight to interact with. And with that, Toda Rabah.